Mass spectrometry is the most accurate analytical tool used for mass measurements. Here, the sample is ionized, separated through mass-to-charge ratio, and detected. The final output is a mass-to-charge ratio spectrum, shown as a plot of intensities or relative abundance against the mass-to-charge ratio. There are different ionization methods that may be used to ionize and vaporize samples. For biological molecules, the methods used are softer, as the samples may be denatured using methods involving high heat. One of the most common methods used for biological samples is electrospray ionization or ESI. ESI is a non-destructive method of ionizing molecules that works by using a charged nozzle to ionize the analyte and spray them as microdroplets. As the droplets accelerate from the nozzle, the solvent evaporates, leaving only the molecule of interest along with the concentration of ions. The result is a spectrum of multiple peaks. This type of ionization has two modes, positive and negative, which may be used depending on the molecule being studied. For proteins, the usual method for detecting molecular mass and amino acid sequence is using positive ionization ESI and tandem mass spectrometry. ESI separates molecules based on a magnetic field. The strength of the interaction depends on the charge carried by the molecule and is in turn quantified into a mass-to-charge ratio. These ratios define portions of the protein, each portion being composed of different mass-to-charge ratios. Understanding their interrelatedness allows for the determination of the protein's molecular weight. On the other hand, determining the amino acid sequence involves multiple MS techniques and is called tandem mass spectrometry. Here, digestion enzymes are used to fragment the protein before being resolved using MS, from which the fragments are further fragmented into ions. These smaller fragments are then analyzed by MS to determine the amino acid sequence. To further understand this, the experiment today aims to 1. Understand the principles behind mass spectrometry and ASI. 2. Determine the molecular mass of compounds using the mass spectrometry spectrums. And 3. Determine the amino acid sequence given the mass spectrometry spectrum.
We begin by looking into four cases as listed here, and make an attempt at determining their molecular weights. For the first case, we look into an ESI spectrum of a small organic compound, where we see a singly charged protonated peak at the mass-to-charge ratio of 299.16. Since it is singly charged, we subtract the mass of the singly added proton to get the molecular weight of the compound. The formula that we can use is shown here. Using this, we get 298.152 daltons. Next to the 299.16 peak are two more peaks. These correspond to the isotope peaks or isotopomers. If we know the molecular formula of the sample, here given as C18H22N2S, then we can calculate the theoretical molecular weight. Using the given atomic weights and the molecular formula, the molecular weight should theoretically be 298.1449 daltons. And comparing this with what was obtained from the spectrum, we see that it is accurate. Moving on to the second case, we now look into a protected amino acid. To calculate the molecular weight of the sample, we use the same formula as the first case and get 452.292 daltons. Notice, however, that we use the 453.3 m/z peak to solve the molecular weight. The peak to its right, which is the 475.2 m/z peak, corresponds to the sodium ion adduct. We were able to deduce this since the difference between the m/z ratios of the two peaks is 22.
We now move on to the positive electrospray ionization spectrum of a mixture of two peptides. As you may notice in this spectrum, two clusters of ions may be seen, one at mass-to-charge 461 to 462, and another at 574 to 576. It may be seen that the clusters at 574 to 576 are singly spaced isotopes, indicating that they are single-charged and have one proton. On the other hand, the cluster at 461 to 462 only have an isotope spacing of 0.5, which suggests that the ion is doubly charged, with two protons present. We will now determine the molecular weights of the peptides. For the peaks found at 461 to 462, we use the same formula as previous cases. However, the number of protons used was 2, as was explained previously. The molecular mass computed is 920.784 daltons. As for the molecular mass of the peaks found from 574 to 576, we use the same formula but with n equals 1, and get the molecular mass of 573.292 daltons.
Moving on to the fourth question, the figure on the screen is the mass spectrum of a small protein. The peaks have been labeled with their m/z values, and the charge state of each set of peaks is also indicated. The molecular weight of this small protein is determined through using the equation we have earlier, where m/z is equal to molecular weight plus n times 1.0079 all over n. Since the mass-to-charge ratio is labeled along with the number of protons per set of peaks, we can substitute the values into the equation to get the molecular weight of this small protein. Upon substituting, the molecular weight was found to be approximately 3,989.96 daltons.
Aside from molecular weight of the peptide, we can also determine the amino acid sequence using mass spec data. So for problem number 5, the spectrum shown is a tandem mass spec product ion spectrum of a 14 amino acid residue peptide. The peptide was generated by trypsin digestion of a protein, and the molecular mass of the peptide is 1569.7 daltons. So for the first question, what do the ions at mass to charge ratio 175.12 represent? So this Y1 peak on 175.12 represents the C-terminal amino acid, which can either be arginine or lysine, since the digestion used is trypsin. Looking at the table on the right, it shows that it's a C-terminal arginine, since if it was lysine, it would have a mass to charge ratio of 147 instead. So for the second question, what amino acid sequence does the series of ions at the following ratios infer? So by subtracting the lower values from the succeeding values, the following sequence can be determined, which is valine, asparagine, aspartic acid, and asparagine again, and 2-glutamic acid, glycine, 2-phenylalanine, serine, alanine, and arginine. So a helpful way to verify the presence of some of the amino acids will be to observe the ammonium ions at the low end of the spectrum. That leads us to the next question: what do the ions at mass to charge ratio 72.08 represent? So the answer to that question is that it is the ammonium ion of the valine, which verifies that there really is a valine amino acid in the peptide. For the fourth question, what are possible amino acid combinations that would account for the mass difference of 186? So by combining the masses of different pairs of amino acid, it was determined that the possible pairs are alanine and aspartic acid, or glutamic acid and glycine, or serine and valine. For the last question, why is trypsin commonly used for protein digestion when mass spectrometric detection is involved? So trypsin is commonly used because, first, its activity is high, ensuring that the specific sites are all cleaved, and 2, its specificity is high, where it cleaves only the peptide bond beside the carboxyl group of arginine and lysine. Here's another problem where we need to determine the amino acid sequence from the mass spec data of a tryptic peptide with a mass of 938. And through subtracting the ratios from each other, the following sequence can be determined, which is phenylalanine, glycine, phenylalanine, proline, valine, cysteine, isoleucine or leucine, and arginine. And as shown in the upper right, the ratios account for both the B and Y peaks, or basically the left and the right side of the peptides after the peptide bonds are cleaved.
Electrospray ionization, or ESI, is a very gentle ionization technique in mass spectrometry and has been commonly used to measure protein masses and investigate protein conformations and interactions. In the following slides, we will be showing studies that utilized this technique to study proteins. In this study, mass spectrometric identification of SARS-CoV-2 proteins from gargle solution samples of COVID-19 patients by Illing et al., the team has successfully developed an ESI-MS diagnostic method that can detect SARS-CoV-2 proteins in highly diluted samples, such as a patient's gargle solution. From the results of the experiment, the authors were able to identify two unique peptides originating from the SARS-CoV-2 nucleoprotein in two of the three samples. It was estimated that the viral load of the two positive solutions were around 100,000 to 1,000,000 genome equivalents per microliter, whereas the negative solution was only around 1,000 genome equivalents per microliter. The peptide sequence shown in the figure originated from the SARS-CoV-2 nucleoprotein that was determined using ESI-MS methods. In the study, detection of native proteins using solid substrate electrospray ionization mass spectrometry with nonpolar solvents by Hu and Yao in 2018 first demonstrated the detection of proteins in their native conformation using ESI-MS with nonpolar solvents such as N-hexane. Nonpolar solvents have low boiling points allowing for rapid evaporation causing heat absorption, reduction of ambient temperature, and condensation of water on the substrate surface. Nonpolar solvents allow the spray ionization to occur at lower temperatures and with fine plumes, which are believed to lead to detection of proteins in their native states.
Fructose biphosphate aldolase is an enzyme that catalyzes the formation of FBP from glyceraldehyde 3-biphosphate and dihydroxyacetone phosphate. The class 1 FBP aldolase found inter alia in E. coli uses an active site lysine to form a shift space with DHAP as part of the reaction mechanism. Mass spectrometry has been used to confirm the amino acid sequence of the FBP aldolase and to identify the position of the active site lysine. From here, the calculated mass of the FBP aldolase is 37,964.3 daltons. The mass measured by mass spectrometry was found to be 37,979.3 daltons. DNA sequencing of the cloned gene revealed two changes to the reported sequence. Val 191 was replaced with leucine and isoleucine 308 was replaced by asparagine. The mass spectrometric data indeed confirmed the changes. Valine and isoleucine have both residual masses of 99.07 and 113.08 daltons respectively. Changing the amino acids with leucine and asparagine, which have residual masses of 113.08 daltons and 114.04 daltons respectively, will result in achieving the mass that is actually measured by the mass spectroscopy. Basically, this is achieved in subtracting and adding the replaced amino acids in the FBP aldolase as shown on the equation below.
Calculate the expected overall mass difference of the derivatized product from this reaction after sodium borohydride reduction. The theoretical average atomic masses are: carbon is 12.0110 dalton, hydrogen is 1.0079 dalton, oxygen 15.9994 dalton, and phosphorus 30.9738 dalton. The mass difference can be calculated by subtracting the mass of lysine from the derivatized product. Based on the given reaction mechanism, we can obtain the molecular formula of the derivatized product. From this, we can compute the mass difference, which is about 154.0321 dalton.
The modified class 1 aldolase was digested with cyanogen bromide to give a mixture of peptides which were purified by HPLC. Mass measurements of the HPLC fractions indicated that the fragment containing residues 194 to 239 of the aldolase contained the expected modification. The isolated fragment, however, gave 4 lysine residues. In order to determine which is the active site, amino acid sequencing from N-terminus was conducted and it eliminated lysine 207 and lysine 208. Since it did not reach the other two lysines, two mutants were created, one with K236A and other with K238A, and their molecular masses were confirmed by mass spectrometry. Both were treated independently with DHAP and their masses were measured again. The mass spectrometric data for the two mutants together with the wild-type aldolase are summarized in the table. Using the wild-type aldolase as basis, the aldolase mutant that reacted with DHAP is K238A since there is a change in its mass after the reaction. K236A on the other hand did not undergo any changes upon its reaction with the DHAP. Following this reaction, the specific lysine that is responsible for the enzymatic activity of Cas1 FPP aldolase is the lysine located on the 328 residue of the isolated fragment.
So just another method of determining which lysine is the active site in the fragment 194 to 239. Include some mass spectrometry in your analysis. The lysine 238 active site can similarly be determined by conducting C-terminal sequencing instead of N-terminal sequencing coupled with mass spectrometry. In C-terminal sequencing, carboxypeptidases are used to sequentially remove amino acids from the C-terminal, which can then be analyzed using mass spectrometry to determine the sequence from the mass-to-charge values obtained. Similar to the given, the weight of the wild type and the mutant can be compared to determine which lysine has been derivatized.
There are some sources of error to consider when doing mass spec in proteomics. One error may be caused by experimental parameters like outdated calibration coefficients and temperature changes during measurement. Another is that the properties of the pre-analytical sample may also be highly varied, along with the digestion efficiency and any preparation-induced modifications, and these could also affect the accuracy of mass spec. Mass spec also has several limitations as listed here. First, varying physicochemical properties like molecular weight, hydrophobicity, and basicity of the resulting proteolytic peptides could affect the efficiency of transmission and fragmentation, thereby affecting the ESI response. In this regard, the better ionizing peptide generates a higher ESI response than others. Also, ion transmission efficiency remains to be a barrier, especially when measuring low-abundant peptides and small populations of cells. And stemming from this is the low proteome coverage, since some of the clinically relevant proteins in plasma have low concentrations, for which mass spec may not have the required dynamic range of detection.
In conclusion, electrospray ionization coupled with mass spectrometry is a very gentle ionization technique that can be used in the study of biomolecules. The underlying principles of the method have been discussed in this report. In the report, two studies, namely the detection of SARS-CoV-2 proteins in COVID-19 patients' gargle solutions and the detection of proteins in their native states using ESIMS and nonpolar solutions, were discussed. Other than protein detection, ESIMS can also be used to detect and analyze inorganic materials and even oligonucleotides. The report also showed how to analyze the data generated from an ESIMS analysis to obtain the mass of a peptide. And lastly, we were also able to show how to determine the sequence of a given peptide given the mass-to-charge ratio values of the peaks.
To further explore mass spec in terms of proteomics, the following are recommended. Assessing the accuracy and efficiency of mass spec against other protein quantification methods, like in using antibodies, gel electrophoresis, and radiochemical labeling. Exploring the effect of varying the properties of peptides that affect the ESI response, and investigating the effects of the complexity of the starting sample on the efficiency of digestion with proteolytic enzymes and how this affects mass spec analysis. Here are our references, and thank you so much for listening.